do with animals. So how did we do this? We just tried to detect recombination, and we did this by looking at what's called homoplasy. And so homoplasys, you probably always drawn phylogenetic trees, I've heard people use these terms. These are characters that are not shared by common ancestry. So I just showed this on the tree here with this base T. If you were to line some sequences, you can see there are very different parts of the tree have the same base. So basically this could be transferred from here to here, and it looks like a recombination event, as opposed to it being ancestral and then lost or all these individual mutations. So we go through all these phylogenetic trees, and we count homoplasies in these trees, and we see these as a pretty robust signal of evolution. For the entire genome. So our technique was really simple in doing this stuff. We downloaded all the sequences from species from a GenBank of that. We took away closely related genomes. We defined the core set of genes. Those are genes that are shared by all the taxa in there. We compute genomic distances. We identified the number of homoplasic sites there are versus the mutational sites in that, and we have a definition of how to apply homoplasy. And we define this one statistic called H/M which is homoplasic versus mutation for every phylogenetic tree. And so this is the only confusing slide that I'll show today, but this is just basically how we calculate this. And so again, the cartoon showing that we have a lot of strange genome sequences. We first sample 4 strains of it, taking them randomly, make a phylogenetic tree. We count the homoplasies over mutations. We do that 100 times. We do that for 5 strains. We do that all up to n-2 subsample strain. And you would see a curve that looks something like this. It's finally asymptotes. It has an H/M value. It's usually less than 1, but there's a lot of homoplasy going on versus mutations. If there happens to be a second species in there, for example, I'm just showing by the red dot, when you sample 4 strains, you're not going to get it. So the curves are going to sort of be identical for the two. But once you start getting that one strain that doesn't recombine, you're going to see this precipitous drop in the distribution. So we look for these discontinuities, which tells us there's probably more than one species in there. And so we did this with 93 strains. Our results is that the species fall into 3 main categories. We have true biological species, which follow what I've shown you. We have cases that are clonal, that is, things don't recombine at all, so H/M is really low, so we can't classify that as a biological species. And then there's cases, like we're called a variant here, where the named species that's in GenBank has sort of this discontinuity in it. We just figure out which strains are from a different species. We take them out, we rerun the analysis, and we're happy found way again. It's now one species, and we have some strains of another group. By doing this, we're able to classify 85% of all named bacteria into species based on the biological species concept. So even though bacteria are asexual, you can apply a biological species concept and apply real species of that. We wanted to see whether the test was robust, so we did it with sexual organisms. We did it with Drosophila melanogaster, with some simulant sequences in there. Again, we get the discontinuity, we take out the simulant species, and we get the same thing. We've done the same thing with humans and chimpanzees in Chromosome 21. Homo sapiens, together you find again this nice discontinuity. You take out the chimpanzee sequence, you get a nice smooth curve of that, which is nice because we know that humans started to breed with chimpanzees and we're separate species. So when we look at that, then we can find out what the limits of diversity there is within a biological species. When people look at 16S sequences, say that 97% identity constitutes a species. However, we see cases where there's a biological species, so that's where the strains in there differ by 30% in the genome. So using that one strict cutoff is not useful at all and doesn't really apply. Here, the Drosophila Homo sapiens is showing you that we're all very closely related to one another. You differ from the person next to you by about 1 base pair out of every few thousand. So now that we can define biological species of bacteria, we're combining this with which is the genome taxonomy database, and what they're doing is they're trying to classify things at every other taxonomic level. So are there new phyla when you find a rare sequence? Is it a new phylum, order, class, and they're just normalizing the distance within a taxon. And now we'll be able to define and classify microbes at every taxonomic level. So if you find an unclassifiable sequence, if you have a genomic sequence of that, we'll be able to figure out what it is, whether it's a new species, new phylum, order. So just some general conclusions from what I told you today, and some unresolved issues as we want to think about the future, is host phylogeny, that's differences that occur during the diversification of hominids, is a primary factor by which ape microbiomes are sorted over evolutionary timescale. We know lots of other factors are going to be introduced in diet, geography, and so on, but there is a large phylogenetic signal to microbiome. And so we can ask, will this limit our ability to alter microbiomes, knowing that there has to be a particular set of microbes that are in there. Chimpanzees harbor these enterotypes that are analogous to those in humans, and hosts shift among enterotypes over their lifetime. Are enterotypes just stable microbial complexes, or do they affect the host types? Do they have some function at all? Bacterial cell groups are co-diversified with great age over the past 15 million years. I'd like to know, do bacteria and which strains persist? What taxa are co-diversified? And are they co-evolving? Is there a function to them? Bacteria reproduce clonally, they differ in the propensity for recombination, therefore we can define true biological species based on barriers to gene flow. And so, is there a uniform and rational approach to delineate organisms across all domains of life? And finally, don't toss out, ignore, underreport those unclassifiable sequences. I tend to think that they're some of the most interesting bacteria that are sitting in microbiome samples, and people usually don't report them. And I'm asking what are the sources, the origins, and the roles of these cryptic lineages. And so finally, I'd just like to give a few acknowledgments to that. The main shout out in the first study was to Mike Warraby. Of course, I'm very, very grateful to Beatrice Hahn, who's been a collaborator throughout most of these studies, and a couple terrific postdocs and graduate students, particularly Patrick Degnan, who worked on copy chimpanzees, and Andrew Muller, who's now moving to Cornell, has done work on the later chimp microbiome and co-diversification. And finally, I'd like to thank the generous support from the National Institutes of Health. Thank you very much for your attention.
So we have at best kind of an operational definition of what species is. Could I just ask you to speculate or amplify on what are the consequences of that for human microbiome research? Just negative or positive?
Well, the main thing in knowing whether something is a species, whether it can be combined and take new genes and intercept, we're finding all these taxa, at least as far as we're concerned, that are co-diversifying, co-evolved, and we don't even know what they are, whether they are new species or just part of a genus or whatever. Some of them are probably functionally important. So we, of course, we always talk about different taxonomic groups are there and whether they are important. Now we can actually, it's more than just human construct and identification purposes to tell you whether genes have been exchanged between a group or not.
Hi, presentation. I think it's really interesting. Can you hear me okay?
No, I can't. Okay.
I think it's really interesting to observe these differences in the composition of microbiome by different species. Have you given some consideration about how this might affect the translational relevance of mouse model research?
The honest answer to that is I've never given it a thought. But if you give me a second, I haven't thought about it at all, I'm sorry to say. Okay, you might.
I think some of the differences, I like what you're doing about the species classification and recombination concept is good with me. But some of the cross feature because geographically they're not in the same area that is they're capable of recombining, although they're not. You know, you could envision something being isolated just because they're physically isolated, but if they wind up getting put together, they might start recombining again. And well, what are your thoughts about that?
Right. Right, so basically, it's the same question you always have. We have squirrels that are on both sides of the Grand Canyon. We have no idea whether they'll ever reproduce. So, of course, this is just our definition of how we would define species. We can't set up every potential mating in that. So there are geographic factors in that. We tend to think that in these cases, things accumulate enough genetic differences through time, and eventually these things are simply say species are separate species of that. But it's always about classifying species to be geographically separated groups, whether they are part of the same or different species in that. We're hoping that bacteria are widespread enough that actually, they can get worldwide. We can actually see whether there's recombination within each group. And these, we just take all the sequences that are named of that group, so it's based on metabolic character, that are named in GenBank. So some of them are from large geographic areas, but some again are from very restricted places. But that will always be about the classification of species.
Maryland, who's going to talk about management and integration of the IHMP data. Thank you. Thank you, Lita. It's really a great privilege to be here today to tell you about the goals and accomplishments of this wonderful microbiome program. This was a 10-year investment by the Common Fund, and the goal is to characterize and catalog the microbes that are on and in our healthy human bodies, and also to enable others to investigate the role of the microbiome in health and disease. And as one very indirect indication of the success and the impact of this program, NIH funding from the institutes has gone up 40-fold since the beginning of the program. This is part of that portfolio analysis that we introduced from the team at WG, so thanks for that. Common Fund programs are intended to be short-term catalytic programs with broad and transformative impact. So what was it about microbiome that convinced the NIH leadership that there should be a microbiome program in 2007? Well, we knew that there were very many human pathogens, and we also knew that they were outnumbered on Earth by more beneficial bacteria. We knew that on our own bodies there are more microbial cells than human cells, and we knew that those microbial cells contained about 100 times as many genes as our bodies do. And we knew that there were indications that these microbes influence our health somehow. But we didn't know what the microbes were that made up the healthy microbiome, and we didn't know how changes in the microbiome influenced our health, or whether they directly influenced our health. But by 2007, we had advances in technologies that would help us answer these questions. Namely, there were faster and cheaper DNA sequencing techniques, allowing very high depth sequencing of many different samples that could be done, and there were faster and cheaper bioinformatics tools to analyze the complex mixtures of genomes or metagenomes from the body sites. So NIH approved through the Common Fund the HMP to go forward for an initial phase funded from FY 2007 to 2012. In the first phase of the program, it is now known as HMP1. Its goals were to catalog those microbes that inhabit the healthy human body, examine whether changes in that microbiome can be related to changes in health and disease, and to provide a standardized data resource that would enable the community to study the human microbiome. It also, along the way, developed new technical approaches, including computational tools, to help generate the data and analyze the data. The data were released as